So today I'll be talking about causal networks. So as you notice, I have a longer title, so we'll eventually go to genomic data. But in the meantime, as you will see, we will talk quite a bit about information. So actually the previous talks that we have, especially this morning, are very nice introductions to the sort of theoretical framework that we will use. So as you will see, most of the talk or the lecture is methodological. But eventually we'll go to real data. And as you will see, the method is rather general, so we can hope to apply it to all sorts of data, mostly biological data, because this is what we are interested in. But I'll try to show you examples of biological networks that we'll try to reconstruct from these methods at very different size and time scales. So we'll go from single cell data, where you basically measure the expression levels of a number of genes in those single cells. And for instance, with a few thousand cells, single cells, you can hope to reconstruct regulatory networks by just looking at the correlated expression of certain of those genes. And we'll look at this example on the differentiation of hematopoietic cells. If you go, and this process basically takes a few hours or days at most. So that's the sort of time scale for this first example. Second example concerns not single cells, but like organs, whole organs, or even a whole organism. And in that case, we'll try to look at or build some networks of gene mutations or gene expression level changes due to in this particular example, cancer. So we'll look at the tumor within an organ. And the sort of samples we have is in the 1,000 also. And the time scales in that case will be months or years even for the small tumor to get bigger and get hopefully not too serious consequence. And hopefully we'll be able to treat it, although we'll see in these examples that there are certain gene alteration that make it much harder to treat, in fact. At even longer time scales, so here we are basically at the time scales of the, within obviously the last span of an individual. If you go to much longer time scale, you can even try to look at data on evolutionary time scales. So here we'll go to millions of years, hundreds of millions of years even. And here we'll try to build some networks will be related to the evolution of the gene blueprint of certain organisms. And in particular, we'll look at the vertebrates. All right. So, okay, let's start from the very beginning. So, especially as physicists, we often have this initial worry about this sort of inverse approaches where, from some data that would look like this, where you have, in the columns you have, for instance, the genes that I was just mentioning. So you have P different genes, and you'll have some samples which will be, for instance, in the example of a single cell expression, that it will be single cells, so cell 1, 2, 3, and you have n cells like this. And from this sort of big data matrix, you hope to be able to build some graphical representation that capture the connection between certain of these genes. So obviously, in this sort of data, when you have, if you pick two genes at random, hopefully, if there is something interesting, you'll find some correlations. And basically, everything is correlated with everything else, but obviously, the name of the game is to try to build some sparse representation of this data in the form of this network, where you where basically you want to capture with direct interactions, even the indirect effects. So for instance, in this cartoon, we see that gene 5 and 7 will be correlated for essentially two reasons, because G1 is presumably a regulator of these two guys, and also a G4. But they don't have any direct regulation between them. So the hope is to be able to find out that there is no direct correlation between G5 and G7, although obviously in the data they will really appear correlated, and that's because there is some sort of common cause in this example. So the worry I was mentioning is that in principle, to build a network like this, if you have p genes, say, well, as you know, you expect to have p squared possible connections. So that's a lot of parameters in principle. And as we know as physicists, as soon as you have a small model with 3 fitting parameters, we can basically fit anything. So now we're not talking about 3 parameters, but more like in the hundreds or thousands, 100,000 parameters. So obviously, it looks bad from the physicist's point of view. So I'll try to convince yourself that things might not be as bad as it looks. And the reason is that actually most of these parameters, as I just outlined, we want to show that their value is 0. So basically, there is no parameter between G5 and G7. So we want to find some sort of sparse representation of this network. So we have much fewer parameters than it initially looks. So I'll try to convince you that this is indeed the case. And for this, let's start from something that is very general. So if you're given this data that I was showing you, so for all the applications that will have, the data will basically look like this: bunch of genes or properties that you want to be the node of your network, and then lots of samples. Hopefully, lots of samples, because the more samples you have, the more obviously precise your reconstruction will be. So the first thing that you can imagine doing is to look at the correlations between these expression of genes. And to do this, you will build, let's say, deviation from the means, and you look at the variance of this expression and covariance between different genes I and J. So you'll build this well-known variance-covariance matrix. And as I said, typically, all these coefficients, they are non-zero. So here is an example of this of a correlation matrix that I made up. And my question for you is, can you guess what the network, the underlying network is? So obviously, so you notice that this matrix is symmetric. That's from its construction, but you also have another symmetry with respect to the second diagonal. So anyone has a guess for the network?
Linear chain.
Good. So that's the physicist's favorite object. So a line of bead and springs. So so I pick up this example because basically we'll do for once the physicist exercise to go from this model to this covariance matrix and find out, how this model, which is obviously very sparse because you see that there are only springs between neighboring beads. So most beads obviously will, the motion of these beads will be correlated through the chain. So that's why this is non-zero. But obviously there are only a direct contact between neighboring beads. So this is a simple exercise that we can just go through if we just write out these equations, Langevin equations, basically for our spring and beads line. I'll add an inertial term, friction term, and these springs, plus some noise. Just to keep it simple, I just put no mass and then just take a unit value for all these parameters. So obviously, this is simple, and that's the reason why I pick it up. You now see that we have these very simple linear equations that we know how to solve. And this matrix, so capital X now is the collection of all the xi. So that's a vector, and L is a matrix, which is called the Laplacian matrix, which was related to the adjacency matrix. So that's how our system look like when we start from these first principle equations. That's the only first principle equations that I will show, basically, and then we'll do the reverse problem. So the nice thing with such a simple system is you can actually solve it. So that's the result. And from this simple result, we can actually explicitly build this variance covariance matrix. Okay? So that's the calculation that you can refer back to if you'd like. And the bottom line is that at the end, once you do this integration, so that's matrix, exponential of matrix, so it's just the sum of these powers of matrix L, this Laplacian matrix. And then when you solve it, you end up with something like this, because we only have power of L's and inverse of L's that commutes. And at the end, for a very long time, and in the infinite time limit, what you end up with is that this covariance matrix is just the inverse of this Laplacian. So if we're given, so now forget this simple model, just imagine we're given the previous matrix. So we're given this. So to find the adjacency matrix, basically, we just have to invert this sigma matrix. And if we do that, so we find this inverse of the covariant matrix, and obviously, it fits what we put in. So the off-diagonal term, they are just the adjacency matrix with a minus sign in that case. And it's full of zeros, so that's obviously as we know because we put it in. But you see that in general, you may hope that even from correlated data, the underlying model is rather sparse. So that's what this illustrated here. More formally, in fact, all these coefficients of this inverse covariance matrix are known to be related to the partial correlations between those variables. And those partial correlations, they are related to how these variable I and j vary when you hold fixed all the other variables. So if I take my chain of beads, now I might want to see how these two beads covary, how their position covary. But I do this by fixing all the other beads. So obviously it's clear that if I look at how these fluctuations due to the noise are correlated to this fluctuation, if I fix the middle bead, there is no correlation between the two, because fixing this bead just screens off the fluctuation between the two, so I get a 0 in my metric. There is no, okay. Obviously, if I pick I and j that are near beads, fixing the other beads won't screen the connection between these two, so it will just give me this agency 1 in my off-diagonal term. So actually, this is a very old trick that I think anyone should know in this field of inverse problems. But there are obviously limitations. So obviously, you need to be for a linear system like our springs and beads, and also this result is not correct if your problem is causal. Okay, this is not causal. I mean, there is no asymmetry between the bead on the left and the right. They are just interacting objects. If now the system is causal, then all this actually breaks down. So let's try to see why it breaks down. So just imagine that we have 3 properties, x, y, and z, which corresponds to 2 independent causes. So x and y are some independent causes for z. So just to be concrete, let's z be the reason or the fact that some of you were late for the first lecture. So either z is being late or not being late for the first lecture. Obviously, there might be a number of causes for being late at the first lecture. X is you couldn't wake up, okay? And UI say you just met a very old friend in the street, when you were coming in for the lecture, and then you decided to go, take a nice French restaurant instead. So there are 2 independent causes. I mean, there is no reason that the fact you meet some old friend in the street and the fact that you couldn't wake up in the morning as related. Okay, they are just independent causes, but both of them will make you arrive late or even miss the first lecture. So now, just assume that we observe this data x, y, and z. We don't know that the underlying model is causal, so we can just apply the previous trick. Okay, we just inverse our correlation matrix, and what we will find is that we will just build some artificial connections between x and y. So the reason is that when I condition on z, so if I hold this variable fixed, so when I do that, I ask myself, okay, given that you were late at the first lecture, what is the correlation between these 2 variables? If you were late, well, so there must be some cause. Either you couldn't wake up or you met this old friend. But you see that if one cause is, if the real reason is because you couldn't wake up, then somehow it's very unlikely that you also met some old friend in the street, and vice versa. So in that case, fixing this condition will build anti-correlations between these 2 variables. Or maybe you were not late, so maybe we should consider the fact that you actually made it for the first lecture, and then you realize that if you actually made it for the first lecture, well then, most likely you wake up early enough and you didn't meet anybody in the street. So obviously it will just build also correlations between these 2 variables. So this problem makes the whole very simple and nice approach of inverting the covariance matrix not appropriate for causal networks. But still, in general, this is a very nice method, very simple. So if you are given some data, it's probably first a starting point to look at this data and see where you can go from there. Okay, so now we are faced with this problem of causation, and that's a cartoon that just illustrates the sort of problems that we are facing. So the point is that we all do it every day. You know, we all manage somehow to distinguish causes from non-causes, from consequences, and so on. And that's not completely trivial. Most of it is due to our past experience. So we have some prior that tell us that some things can be the cause, some other things cannot be the cause, and so on. But we basically manage to do it quite well on the daily basis. So now if you're given just the data without any prior, it's much more difficult. In fact, we can even ask ourselves whether this is even possible, because I'm sure all know that correlations do not imply causation. And one clear example of this is between the barometer and the weather. Obviously, these two observations are hopefully correlated. But in fact, none is the cause of the other one. Even though the barometer is supposed to give you some signal before the weather, so even this time delay between the two is not enough, obviously, to make the barometer the cause of the weather. So in this simple example, we know that, in fact, the correlation between these two observations, the barometer and the weather, is due to a common cause of the two, which would be some atmospheric pressure, let's say. So the very intriguing observation and result now is that if you go beyond 2 points, so this fact that you cannot predict causality from correlation is only strictly correct for 2-point correlations. So if you're only looking at 2-point correlations, this is hopeless. You will never be able to distinguish between cause and consequences. But the rather striking result is that if you look at more than 2 points, so 3 or more points, exactly like the examples I was showing you earlier, if you go beyond 3 points, then you might get some trace of information in the data. So one of the first to think along this line is Hans Reichenbach, who proposed some principle that we could call the common cause principle. So basically his idea is that if you observe two properties, E1 and E2, or events, two events E1 and E2, that have no reason somehow to be correlated a priori. If you observe that they are correlated, then your intuition tells you that there must be some common cause that explain these two unlikely coincidence of these two events, E1 and E2. As we will see, so this is actually a nice intuitive idea, but as we will see, it doesn't really work out when you really want to analyze real data. So really, the only graphs that has some causal information in it is precisely these graphs that I was showing you before. And this idea is due in particular to this man, Judah Pearl, who showed that, in fact, this is the so this principle, we could coin it, this is not how it's coined typically, but we could coin it like the independent cause principle, like the one we outlined before. And so the idea is that if we have three properties, and we can observe that these two properties are correlated, and these two are correlated, but somehow these two are not. So if we look at the correlations, if we build our correlation matrix, we find that x and y are not correlated, and yet both are correlated to z. Well, then what it tells you is that z cannot be the cause of either x or y. Because if z was the cause of x or y, then somehow, so if you had an arrow like this, well, then somehow you'll be correlation between these two. Okay? So the idea of Pearl and others, so that's, we're talking about the late 80s, is that when you look at a correlation pattern between three observable x, y, and z, where two are independent while they are both correlated to a third variable, basically the idea is that the only consistent graphical model corresponds to these pointing arrows. And these pointing arrows really basically tells you that z cannot be the cause of ui nor x.
So you still look at 2 points correlation in this picture?
Well,
X, Z, Y.
yeah, I'm just describing this 3-point object in terms of 2 points. You're right, but in a second you'll see that we actually look at them in terms of 3 points as well. Okay. Okay, so now I'll try to give you a brief review of how, basically the two main approaches that have been developed since the late 80s, early 90s to actually reconstruct those causal networks. So the first method, which is actually the most used, is this Bayesian inference. So you can call them search and score methods. The whole idea is to basically sample the space of networks over your observable. So if you have only 4 observable in this cartoon, well, then maybe one possible network, causal network would be this one. And then the idea is that you have a way to estimate the score, the likelihood of this network. Okay, so this is the likelihood that we will introduce later on. And then the idea is that basically, ideally, you will like to look at all possible networks and just pick the one with the best likelihood, the highest likelihood. So it works. The problem is that you cannot really sample exhaustively a space if you have more than 10 or so variables. So if you have more than 10 variables or 15 variables, then you need to do other tricks to sample efficiently your network space. Basically, doing some sort of Monte Carlo trick where you jump over network space and accept or reject moves depending on whether you improve or not your likelihood. So you need some sort of heuristics to explore this network space, but actually some very powerful heuristics have been developed, so it actually works quite well. Probably, in my views, the main limitation of this approach, so you have this network space issue, which is maybe not too much of an issue. The main drawback, in my view, is that you only, these models, they only corresponds to networks with directed edges, so with cause and consequence. And obviously, you don't know a priori. So if you're given some data, you have no idea whether this is a causal or not, or you may not have an idea. So if you fit it with a Bayesian network, the only answer will correspond to this causal network. So it might not actually be the best model, but you don't know how to check this assumption, basically. So the second type of approach, which is much less known, in fact, is called the constraint-based approach. So here the idea is rather different. So we'll start from a complete network. So initially you assume that all variables are connected. So that's the so if the actual data is the same, and we will just assume for the sake of simplicity that the real network is the one that we just found using the Bayesian approach. So this is this simple network. And now the idea is that we will try, so obviously there are two edges that needs to be removed, one between t and z, and one between x and y. So these two edges, somehow, we don't need them. So somewhere in the data, we should be able to find out that these two edges are not necessary. And the way we will find that they are not necessary is by basically playing this trick of holding some variables and see whether we can stop the information flowing from between different nodes. So since we know the answer, this is written up there, we see that basically the first link between x and y can be removed, because if we hold t, well, we just stop the information flowing from x, between x and y, okay? So that allows us to remove this edge, okay? And then the second edge between t and z that we will need to remove, here, in order to find this conditional independence between t and z, we have to hold not only one variable, but actually the two, x and y. So because x is a common cause of the two, and y is this intermediate variable that just allow the information to pass from t to z. But if we hold these two variables, then the variation on t and z become uncorrelated. Okay, so that yes.
From the experimental data, how do you remove this?
Okay, so you do exactly what we've done, except that you don't know the answer, obviously. So the idea in this approach is that initially you assume that all properties are connected, or all genes are connected, and you will try to prune this network to remove as many edges as you can. And to do that, you will, for each pair of nodes, let's say x and y, you will try to find other variables in the network, like t in our case, that somehow explain the correlation between x and y. Okay? And the whole idea of this method is that as soon as you find this condition, so you run some statistical test on the real data, and as soon as you find that somehow condition on t help you pass this statistical test of independence between x and y, then you will remove this edge between x and y. And then you go on like this for all different pairs. So the way it's set up in details is that you first look at, for all pairs, at a single contributor on this indirect path. Then you remove the corresponding edge, and then you increment to two contributor, like x and y for the edge between t and z. Okay, and if you find with two contributors conditional dependency, you can remove these edges, and you go on like this with more and more contributors. So obviously, it sounds not optimal at all, because you need to look at all the combinations of contributors in principle in order to test. So for Bayesian network, like the one up there, you can actually restrict yourself to neighbors of your segment. So if you look at x, or let's say if you look at t and z, you don't need to look at all other nodes in the network. So this network is very small with 4 nodes, but you can imagine that it could be much wider. And so in fact, you only need to look at neighbors of t and z. And since you're pruning this network, eventually each pair has fewer and fewer neighbors. So in principle, you can work out that in fact it converges rather nicely. So the whole algorithm is actually polynomial. If the network is sparse enough, you will just prune your network rather efficiently and end up with some answer. Problem with this is that this is really not robust to the statistical test. So because obviously you realize you start with a complete network, so you have this huge number of edges, and hopefully you hope that the final network is rather sparse, so you'll go to p squared edges to p edges. Basically, you want to remove all these edges, and you'll make errors. Inevitably, you'll make errors, and the problem is that the early errors, so if you remove early an edge that you should actually keep, well, this early mistake will trigger many more mistakes afterwards, because you will need to keep edges that you should remove to compensate for the edge that you removed initially. So although the method is very nice on paper, as I told you, in principle it's polynomial. In practice, there are serious problems, and it's basically not robust to the finite size of your data sets. But let's go through the whole approach until the end, and then we'll see how we can actually improve this method to make it robust to the finite size of the data set. So after we actually prune this network, we end up with a graph that has basically the correct edge if we were robust. So it has the correct edge. We call it the skeleton of the graph, but we don't have orientation yet. Okay. So the orientations we will add them now. And the idea is to basically review all the missing edges. So we just remove x and y. And we remember that we remove x and y by conditioning on t. Okay? And now in our skeleton, what we observe is that there is also z that is a neighbor of x and y, but that was not involved in the removal of the edge x and y. So because z is a neighbor of the two and was not, passing information between x and y, then, using the same reasoning as before, we just conclude that z somehow must be downstream of x and y. Okay? Because if it was not downstream, then in principle it should be in the condition variable too, because it would have contributed to the correlation between x and y. So that's the second step. It's when we orient some of these edges. You see that on top here we have no orientations, and this is because, in fact, as we will see in a second, there is nothing in the data that can really tell us how to orient these edges. So they can even be non-oriented, so they can be non-causal connection between these two guys, unless you add these special subgraphs that we call a V structure. So that's the V structure. So any orientation of these edges that doesn't create a new V structure is compatible with the data, and it's equivalent with respect to the data. Okay? So basically the method tells you that this is the most general model. In principle, there is also a third step in this approach. So once you find the skeletons, or remove the edges, orient some of them corresponding to V structures, you may end up with remaining edges, with an entering arrow. Okay? So these edges with an entering arrow and without edges there, so they're like open triplets. If you didn't find that they would correspond to a V structure, so if we didn't find that there was an arrow pointing to Z from W, well, then we can assume give, you know, if the assumption is that the underlying model would be a Bayesian graph, we will conclude that in fact the arrow is pointing from Z to W. Okay, so this V structure, their orientation can be propagated downstream on the network. If they are not V structure, then they have to be a non-V structure somehow. Yes.
Why not having X and Y projected in W?
X and Y. Well, that's the data will tell you this. So obviously X and Y could project to W, but somehow in this cartoon, okay, this is the real network, remember? So basically we could find out that there was no connection between X and W, between UI and W, say. And we find this by conditioning on Z. Okay? So we could remove this edge. And we could also remove the edge between X and W for the same, with conditioning on the same Z node. Okay? Yes?
Excuse me, how do you assess conditional independence here? Like you get real data.
Okay, so if you're real data, you need to put up some statistical test. So, for instance, some mutual information. We will detail this possible test. And you have to have some criteria telling that, you know, given the amount of data that you have, given the correlations or partial correlations that you find between these two nodes, then you will decide that statistically this correlation is irrelevant given the data. So you have to pass some sort of statistical test. If you have infinite amount of data, then, you know, the answer of this test will be always correct, and then the method can be proven to be correct. Okay, but as I said, typically you don't have infinite amount of data, and so the method will make mistakes. And in fact, because of this cascading effect, the method really doesn't work so well. But still, on paper, this is a very interesting method, which so one of the good points of this method, it's in fact you can recover a much broader class of graph. So we only saw directed and undirected edges for the moment, but we will see in a second that you can also have bidirected edges, which corresponds to latent variable that I will explain in a second. But again, the drawback here is that this is not robust on real data. So basically, this is the reason why most of you don't know this method. Is if you apply it to real data, it doesn't work. Okay, but still, on paper, it's a very interesting method that we can hope to make work eventually. And also, it's fairly slow. Okay, so in order to improve this method, we will use information theory. So the idea is to basically take the best part of these two approaches. So the best part of this approach is that, you know, you can recover a much broader class of graphs. So you don't, for instance, we can hope that if you're given some data, you have no idea what the connections between those variables are. We may be able to predict whether this is causal or non-causal relations between these data, which you cannot do using Bayesian network, as we said. So in order to make this method work, we will basically use some form of score, some form of likelihood. So all this pruning process of the network will be based on scores, so that we make sure that, you know, the early edges that we will remove in this graph are correct. So eventually we'll make mistakes, but we want to make mistakes as late as possible. So the early decisions, we want to make sure that they are most likely correct, and that will be based on maximum likelihood. Okay, before we go to this, let me introduce this much broader class of graph. Question, yes.
I was wondering, suppose that you are able to get the graph, the
Yes.
interaction graph, for a subset of your nodes by ignoring the other parts of the nodes.
Yes.
And then how the interaction graph within the component change by including the others? So I mean, can you split the problem into subcomponent of your system?
Can you split? So if you basically in this approach you start by assuming no split at all. You assume that everything is connected to everything else. If you have prior knowledge that these different parts should be unconnected, or for instance if you know that these two properties are not connected for some reason, you can put it in, and instead of starting from the complete graph, you can start with something that is a bit pruned already. I haven't seen much along this line, but yeah, in principle, this is feasible. You can put by hand some prior, saying, well, I know that this is not but this is an edge that does not exist. I'll just remove it. You can do that. Okay, so this class of graphs that I'd like to introduce comes from looking at this. If you go beyond 3 points, so we look at this object with 3 points. If you go to 4 points, you can end up with what I would call a path of colliders. So basically, I could have the same correlation pattern between these 3 nodes. I could have it also not only between the 3 nodes, but between the 3 nodes too. So basically what it means is that Z is not the cause of X or UI, and UI is not the cause of Z and W. So yet in the data, there is clearly some connection between Z and UI. So the inference is that this connection is due to some other variables, L, that we call the latent variable, that is somewhere that cause these correlations, but that we do not observe in the data. So for instance, it could be a gene, transcription factors that regulate these 2 genes, Z and UI, but that we do not observe. We have no data on it. Okay? But still, from this correlation pattern, we can guess that there is some missing cause between Z and UI. Okay. So when you include all these possible relations, undirected, directed, or bidirected, you end up with a class of graphs that obviously is much broader. And basically, there are the same sort of constraint-based approaches where you start from a complete graph, prune the edges that are not necessary, orient some of the V-structure, that may lead to bidirected edges like this, and then you can also propagate some of these initial V-structure to downstream edges like here. But again, this is not robust. I mean, in practice, it doesn't really work if you use this statistical test to prune this network. Okay. So we will actually need to introduce some information theory here. So the nice thing is that we saw quite a bit already by Stephanie this morning. I will cover very similar things, but actually from a rather different perspective. So I think it will be rather complementary. So we'll also introduce this Shannon entropy. But we will basically introduce it as Shannon did. And the idea is that if you observe, so now we have data. So we have n samples, capital N samples, of this random variable x, okay? And basically we'll assume that these samples come from the same distribution that we don't know, P of x. Then the idea is that if this sampling, if you have enough samples, well then the probability that you have a specific sequence of value of x, so from x1 to xn, this probability will basically converge to something that in fact does not depend on the precise sequence. So, and it will converge toward this exponential minus n, the number of samples, times some function, which is basically the Shannon entropy. So actually, I think it's, because it's so central, maybe it's interesting to see how to prove this. This is very simple. So the idea is that, and so it's called the asymptotic equipartition property. So the idea is that so as you can see in this theorem, there are assumptions, which is that we have to draw these x, these successive x, independently. So they are, each draw is independent, and they all come from the same assumed distribution. Okay, so if they are all independent, we know obviously that the probability that they to get this specific sequence is just the product to get each of those values. And then basically we will build this quantity okay, which for independent value will just be converted to the sum of this log over all samples. So that's simple. So here, obviously, we use this first I in the ID over there. So this is independent independent variables. Okay. And now we'll use the second I, which is identically sample from the same distribution. Okay? So if we use this second condition, basically we can transform the sum over all samples over the levels of this distribution. So for each of these samples, we get 1 over n, and if these guys are just distributed in different levels from 1 to r, say, then we can transform this sum into the values of this x over this number of level r, px, which will be the number of 1 over n, the number of sample that will get the same value, log px. Okay, and that's basically our formula. Okay, yes?
Maybe I missed it, but what's the level?
Sorry, what? Okay, so that's a good question. So a level is the fact that these values are discrete. So our distribution is a discrete distribution. Okay, and that's very important, in fact. So we can give a few remarks. So in fact, indeed, so first thing is that this Shannon entropy, is positive, clearly, because of the sign that's clear. And it's bounded by log r, which is where r is the number of levels, and that corresponds to uniform distribution. And obviously what you see is that this bound, in fact, diverge if you go to continuous signal. So in fact, this Shannon entropy somehow is not well suited to go to the continuous limit. Okay. Okay, so that's the first thing. Okay, and maybe the second remark that I will make, so which goes back to the interpretation of Shannon, in fact. So this is related to this equipartition property. So as we said, so the probability of this particular sequence are all the same, in fact. They're all related to the entropy of the distribution from where they come from. So they're all same probability, okay? But obviously that's what it tells you about is that the typical sequence in the infinite size limit are all identical, are all as likely. So that's the concept of typical sequences or messages. And obviously the number of those typical sequences is just the inverse of their probability. So that's basically the number of typical sequences. So obviously if your alphabet has just one letter, so what it means is that there's only one value possible for x. So P has to be 1, then the entropy is 0. And so obviously the number of sequences is just 1. That's a sanity check. Now, if you draw some sequences from with many different levels, say some proteins with 20 different amino acids, well then, if these amino acids are all as likely, then the probability will be 1/20, say. Obviously, we know that they are not all as likely, so you'll have some intermediate entropy. And if the sequence of this protein is long enough, so basically, the typical messages will all have the same probability. Okay, so now we need to introduce something that was already introduced earlier, which is we're not just looking at the data, we want to model the data, okay? So we want to, because we don't know this P, for the moment we just worked around with this P, but we have no real access to this P, we only have the data, okay? And the idea is we'd like to, assuming that the data come from certain distribution Q, okay? We want to find out how likely it is for us to observe the actual data point, x1 to xn, if we assume that they are drawn not from the true P, but from the assumed theoretical distribution Q. Okay? So this looks very similar, and in fact it is very similar. Let me remove this. So now basically the idea is we'd like to find out the probability not from the true probability P, but from the assumed probability, theoretical probability Q. Okay. And as you observed, so we actually recover this Kullbaum-Leibler divergence that we introduced earlier. So that was introduced by Stephanie just before. So I won't go into the proof, but in fact the proof is very similar. So this is basically the same proof. So this is just a one-line proof for this cross entropy or log likelihood. So you go from, you take the log of this the sequence Q, the sequence x1 to xn, assuming that it comes from Q. If there are independent drones, it's just the sum of all these log Qi. And then in the limit, in the infinite limit, you can just transform this sum over samples to sum over the actual distribution P, okay? And then you basically get the answer. Okay. Okay, so for the moment, I had only one variable. But in fact, this variable, it can be a vector. So this x that I was mentioning can have different values. So in fact, I will basically apply it to my network problem. So now I have some network with different different nodes, x1 to xn. I don't know the network yet. Okay, I have some data on this observable, and I will just write the same thing. And I know that if I draw enough, if I have enough data on this multi-variable data set, then I'll be able to learn something from this distribution. So eventually, I'll need to introduce this multivariate entropy. Between all these different properties of the network that I'd like to reconstruct eventually. Okay? So you have the expression over there. So obviously, if this is independent, if all these properties are independent, as we did just before, the probability, the joint probability is just the product of the different variables. And then, obviously, it will convert into the sum of this entropy, okay? If they are independent. And if we go, so just to make it concrete, if we go to two variables, x and y, so just the sum of these two. And obviously, this corresponds to a network where there is only two nodes and they are not connected. Okay. In general, we'll expect some connection over this node. So basically, they won't be independent, and so there will be a correction to this, which is basically this mutual information between the two. Okay. So I can rewrite it the other way around. So those are exactly the same formula as Stephanie just showed you. I just keep them completely symmetric because, as you observe, they are symmetric. So I think it's easier to see what they mean if you keep them symmetric. Okay, so one important point is that here I added the semicolon, while in the entropy they are just commas. And the reason is that, in fact, you need to distinguish, because these variables can be also multivariable. So this x can be corresponds to two x1 and x2. So it can be a two-node property. And then you see, once you develop this property, you see that you cannot exchange x1 and ui. So obviously you have to keep track of this, and this is why we use these different separation between variables in information. Okay, so that's the mutual information. So, but in principle, you can in fact define exactly the same way multivariate information. So not only 2-point information, but 3-point information, and in fact n-point information, by just keeping the same idea. So you just add the entropy of the single variables, then you subtract the pairs, and then you add the triplets, and so on. And the nice thing with this inclusion, exclusion formula is that you can reverse them very easily. And so the mutual information can be expressed very easily from the entropy, and vice versa. The entropy can be expressed back to the mutual information with basically exactly the same formula. Oops. Okay, so we also need to introduce conditional entropy, which was already introduced by Stephanie also. So I'll just write them again. Sorry, what? Yes. Oops. Okay. Okay, and then then we will need also to introduce conditional mutual information. And so conditional mutual information, they are just basically exactly the same formula with respect to entropy, just that you need to condition on the variable A in that case, okay? And then when you work out the details, you will end up with something looking like this. So, okay, let's take a concrete example. So if I have 3 variables, x, ui, and a, the joint probability between x and y given A is just the product of the two. Okay? Because basically there is no direct link between the two. Okay? So we see that in that case, obviously, this will correspond to vanishing mutual information. Okay? Okay, so maybe we can draw a graph that will maybe help us to remember all this. Okay, so if this represents my whole joint distribution, if these two ensembles corresponds to the independent variable, to the distinct variables, the intersection will be the mutual information, and the rest will be this conditional mutual information. Okay, so one important properties that I forgot to mention is that while for all set of nodes the entropy is positive, despite the symmetry of those formula, in fact, it's only for sets that are smaller than 2 or equal to 2 that the information is positive. So the information, when it goes to more than 2 points, can be negative. So we will see this in soon. Okay, so let me rewrite just one thing. If I can introduce the entropy of A in the last formula, I will just remove all the conditioning, okay? And now you see that I can combine this conditional mutual information with the 3-point information that you have written up there. And when you do this, you end up with a formula that will basically remove the A. So we will find that if we sum this and we sum that, whatever A, it will just drop. So what it means is that we can basically decompose the information between x and y by introducing some extra variable A, okay? And the way we can interpret this is that this is the indirect contribution of A to the mutual information, okay? Indirect, and that's the remaining let's say direct contribution to the mutual information, okay? So now the idea is, okay, so what I can do is I can also write exactly the same equations. So this equation is just the one before last, and the last one is the same equations, just conditioned on a set of nodes AI from 1 to n-1. So I just introduced this conditioning on some other nodes, but that's the same formula. And the idea is that this formula will allow to basically collect progressively the contributions of other nodes to each pair in the graph, okay? So that will be my last formula. So basically I can iterate this formula if I put just one node, this looks like this. Then I can pick another node, A2, condition on the first one, and then I have some rest. Which will look like this. And I can basically iterate. So eventually, the last term, if this becomes equal to 0, my last term, which is a 2-point condition on some set of nodes, when it will become 0, if it becomes 0, it means that I have no direct connections in this graph. And what it means is that I can basically account for all the mutual information in between each of these pairs by contributions from a number of other nodes, A1, A2, up to AN. Okay, so let me move on. Okay, so the idea is, so why are we doing all this? It's because we can express the maximum likelihood exactly in this format. So this Kullbaum-Leibler divergence is basically the sum of all this conditional mutual information for all the missing edge in the graph. Okay? Where the conditional variables are what we call the ancestral variables that are upstream of either x or y. So interestingly, this expression also helps us to estimate, to get some confidence estimate on each of the retained edges. And this we'll do by comparing the probability that this edge is removed by the same probability after randomization of the data. So this is some sort of p-value. So we'd like to have, so the smaller this confidence ratio is, the more likely the actual edge should be kept in the network. Okay. So the idea is we will basically apply this formula to our small graph. So I can go fairly fast on this. If we apply it to our favorite V structure, we see that there is no upstream node of x and y. So this is just a mutual information that remains in the log likelihood. Well, if I look at the alternative structure, this is the conditional mutual information, because there is Z is always the parents of at least one of the edges. But as you realize, all these guys, they are all equivalent from the maximum likelihood principle. So basically, the data is not able to distinguish those, and we may as well remove the orientation altogether. Yet, there is definitely a difference in likelihood between these two motifs. Okay, so we can compare and tell what the data is most likely to corresponds to. So if we look at this difference here, we'll basically end up by looking at the difference between the mutual information and the conditional mutual information. And as we said, this is the 3-point information. Okay, so this has a sign. This is, and clearly, when this 3-point information is negative, the data is more compatible with a V structure. When it's positive, it's more compatible with a non-V structure. And in that case, this formula that we wrote already tell us that Z is, so this 3-point term is basically the contribution of Z. And so if the true graph is this one, the last term is exactly 0. And so all the mutual information between X and Y is due to the contribution of Z. Okay, there is one thing that I should not forget. This 3-point information is completely symmetric, as we can see. So there is a trick here, because obviously these graphs are not symmetric. So we only decided so far whether the underlying graph is causal or not, basically. So we need to choose how to orient it. So we need to compare the different orientations of the different, either V structure or non-V structure. And when you work out the details, you find that the favorite orientation should correspond to the weakest edge that you remove. So the orientation should correspond to the weakest mutual information at the edges that you remove. And this is, it has to be this way because otherwise it would just violate what we call the data processing inequality. Okay, so for the moment, we saw that we need 3-point and 2-point information to learn somehow this graph, but we just look at very small graphs. So now we obviously need to go beyond these small graphs. And to do that, we'll basically use exactly this approach that I was outlining before. And the idea is to collect the best contributors until we hopefully find a vanishing rest. Okay? So basically, the method we can write this equation the other way around. So if we just look at the rest, we'll just subtract 3-point conditional mutual information from the initial 2-point until possibly this rest becomes 0. And then we basically have to rank all these edges with the best score, and then we just prune our network starting from the edge from which we are the most certain that we won't make an early mistake. Okay? And then basically the algorithm is like the classical one, so we prune this network in the first step, and then we will orient some of these edges using the sign of this 3-point mutual information. If it's negative, it has to be a collider. If it's positive, in principle, we can also propagate. Okay, let me briefly show you some benchmark, and then I'll try to give you some result on real data. So the benchmark here show you that, in fact, this method greatly improved the classical approach. So this F-score measures let's say, the accuracy of the prediction. So you want to be as close to 1, and this is the number of samples. So obviously, if you don't have any samples, all method fails, which is not a surprise. But then if you get more and more samples, you see that these curves can become not quite 1, but not bad. The different colors corresponds to the number of hidden variables. So here we have some of these bidirected edges. So there are some variables that we know, but we don't look at them, and we still try to predict this network, and we can still predict this network rather well. So the more you add hidden variable, the worse your predictions are, but they're not, they are not collapsing too fast. In fact, even with 20% of variables that are hidden, we're still able to make reasonable predictions. And this method, as you can see here, they are basically 100 times faster than the classical methods, and they are better or comparable, but with much fewer samples. So you get, using this likelihood, you can get as good or typically much better result with 100 times fewer samples, in fact. So you actually use the information in these samples. Okay, I'll skip this, and let me flash some of the result on real application. Okay, so I'll go to the first application on single cell expression. So here the data, and you'll have a talk on Monday, if I remember correctly, by Leila Perrier about this process of single cells that differentiate into hematopoietic precursor and endothelial precursor. This happens in a matter of few hours. We have 4,000 single cells data. That's from the Godgen's lab. And you have 4 points into this basically one-day follow of this process. When you look at, so this is a PCA, so this is when you look at, you try to find the greatest variance in this data, and that's in the same representation after clustering these data into different populations, because as you can see in the PCA, you observe that there are some variations and there are different subpopulations of cells in this data. In particular, this hematopoietic precursor will eventually pop up at the end, at the very last data point. And before that, you have basically two subpopulations that one, the primitive population will eventually become less prevalent while the anterior precursor will just appear. So when you use the data and build some network, you end up with something like this. So this bifurcation, which means that you have different population of cells in this data, appears most clearly here because through these blue arrows. So the red dot corresponds to genes that are specific to hematopoietic cells, the purple dot to endothelial cells, and the blue dots to all cells, basically, common to all cells. And you see that expression of hematopoietic genes is basically anti-correlated with the endothelial cells, which is obviously expected. Some of the direct contact that we predict have been reported in the literature, especially in this paper, in particular around these three genes, but in other reviews, many of these Icaros targets we actually predict. And as you can see, we have some of these edges that are bidirected, so we predict that they would correspond to missing genes that we think impact the network but are not observed in these data sets. And for instance, for these two pairs of Lil1 and ERG, when there is a plausible candidate, which would be a GADA2, which is well known to be involved in the regulation of these genes, and yet not in these data sets. Okay, another example of application at much wider scales would be this tumor sample. So here we look at breast cancer data. So we have 1,000 tumor samples from the COSMIC database. And so in this database, they report mutations in the tumors, change in expression levels, either under or overexpressions, and also change of ploidy. So I would like to focus on change of ploidy here. So change of ploidy means that certain cells or certain tumors, say, have doubled their genome. So as you can see in this sample, is basically half of the cells have almost ploidy 4, which is double as the normal ploidy. And here on these graphs, you can see that the ploidy has a strong, is strongly correlated with the change in mutations of p53. As you can see, the mutations in all the other genes are essentially negligible. So, and that's explained why we have very few yellow dots in this graph, which corresponds to mutations. So mutations are basically not, and maybe surprisingly not, connected too much with the change in expression, except for a few genes like p53, which is well known. But you see that the ploidy changes has a strong impact on the expression of these genes, but that the reconstruction tells us that this is not a direct effect. So the ploidy is basically only affecting the expression of the other gene through the mutation of p53. And that, in fact, makes sense from the clinical data, because cells that have undergone this ploidy change will be much trickier to treat, basically because after genome duplications, these few cells will eventually mutate and will be a bit different. So because of this redundancy in the genes, you will easily accept many mutations, and you have different subclones that will appear. And then in particular under treatment, so when you treat a patient, this right here, if the cells are basically all the same and have not been able to divert too much, typically the treatment will be effective, and you will hope in many cases that you can in fact, target and remove the tumor. On the contrary, after whole genome duplication, because of this diversity, although you will kill most of these cells, it's actually very likely that there will be some clones that will be resistant, just by chance, of the treatment. And then if you kill all the other cells, well, basically these nasty clones will have all this room and will eventually invade and grow a tumor that will be much harder to cure. So these whole genome duplication events, clearly they have some serious impact on the tumor progression, and that's now studied more and more. But they also have, this whole genome duplication, they also have consequences on a much longer time scale. So here now we are the third application where we're looking at the evolution of species, in particular through whole genome duplication. So in vertebrates, in blue they are all vertebrates, we all descend from this single species that underwent these 2 rounds of whole genome duplications here. That's about 500 million years ago. And although after whole genome duplication you have this huge redundancy of genes, so you have twice as many copies for all genes, eventually over time most of this redundancy will be lost. So basically you only keep 10% of the initial duplicates after 100 million years. What we noticed is that these duplicates that you keep are surprisingly the ones that are the most likely to be the causes of a serious disease, in particular dominant disease. So it's like, you know, you just keep the ones that are the most likely to have a negative impact on your health, which is obviously quite surprising. So we started to look at this problem, and now the idea is we'd like to see how, so we'll try now to build some model from the data that we have. So the data now, the samples of our data will be the genes, the human genes, okay? So unlike before where the genes were the nodes of my graph, now the genes, they are the samples. So I just transpose my matrix. So the genes will be the sample, and my properties, or my nodes on my network will be the genomic properties. So the properties we're interested in are related to disease. So whether, you know, this particular gene is involved in cancer or not, whether it's involved in other disease, or whether this is related to dominant disease or dominant negative effect, being part of complexes, we can pick any different properties here, and we'd like to see whether there is a connection between their disease susceptibility to disease and the fact that these genes have been duplicated in evolution, either by independent duplications, so just one gene at a time, so that's called SSD, small-scale duplicates, or by whole genome duplications that I just mentioned. And in that case, we call them onologues, after Susumu Ono, who first proposed these whole genome duplications. Okay, so probably for the sake of time, I need to be a bit fast, so I'll go to the last slide, the conclusion. So this is this process. You have 2 rounds of whole genome duplication at the base of vertebrates. And then, so here we pick some of these properties that you can observe for 20,000 genes. So the number of samples is huge in a sense, 20,000 genes, and just a handful of properties. And what we could see is that the retention of these onologues is indeed mostly connected to the dominance of the genes, or also some other properties that are related to the autoinhibition of these genes. And although they are, I mean, the fact that you retain some of these genes is connected to other properties, like being part of complexes, for instance, which has been advocated in the field, or expression levels, we can explain all these correlations by indirect path. Okay, And that fits earlier model. So as you realize, this is a data-driven model. So we just look at the data and try to infer some model without specific assumptions on the model. You can also build, population genetics models starting from first principle, like the line of Beeden Springs that we had before. So you can write the same sort of equations for population genetics and trying to figure out what could be the reason for this analog retention. And that's what we did, if you're interested in this paper. And basically these two approaches, either based from first principle or from data without a priori assumptions, somehow confirm and coincide together, which is actually interesting to see complementary approaches agreeing for the same data. Okay, so with this, I'd like to close and thanks the students and postdoc who worked with me on this project, Louis Verny, Nadir Sela, Severin Affelt, Parapriya Singh also, Julia Malaguti, and also now Guido, who joined us recently. And with this, I'm ready for questions. Sorry?
Just before application.
This one?
No, it was before application one. I think the slide before that.
This one?
No, further. It was the part where you have previous methods in this one.
Okay, the benchmark comparison.
Yeah.
Okay.
I didn't quite understand what this was. So you know the structure of these networks completely.
Yes. So those are benchmarks. So you see here we look at 3 different networks. One has only 500 parameters, one 1,000, and 100,000 parameters. Okay, and the blue is the state of the art basically from 2012, based on this statistical test that I was mentioning. And you see that in particular, if you have many parameters, like 100,000 parameters, well, this method they fail completely. We don't get, really up to 1. But what is encouraging is that you see that even, we need obviously a lot of data. So with 50,000 samples, which, in a sense is a lot of samples, we see that, we can still hope to be able to grasp some of the information behind this data to improve our prediction. Yes.
So these actual networks, were they just artificially constructed?
Yes. So they are so-called real life based networks. So basically, you know everything. So they are like Bayesian networks where you know where you can generate some data given the parameters. So that's what we did, and then you can reconstruct by hiding some of this data, and we hide up to 20% of the generative nodes. And obviously, when you go to, so here it's actually not the correct number. It's from 0% to 20%. So when you go to 20%, you lose, 0.1 basically in F-score, but you're still able to reconstruct to some extent these graphs, which will have these directed edges, which corresponds to these hidden variables. Yeah.
Orientation of the arrows was not clear to me. How orientation of the arrows is how do you define it? I didn't understand that.
So the orientation of the arrows comes from this this very first V structure. That was so that's this example of being late at the first talk. Okay. So if you have 3 variables, so you just observe these 3 variables. 2 of them are independent, and yet they are both dependent with a third one. Okay. Then the third one cannot be. So the graph cannot be this one, because with a graph without orientation at all, like this one, then you expect some correlation between x and y, because there is, through z, there is some connection. So these guys cannot be basically independent if they are both connected to z without orientation. Okay? And then the orientation cannot be in the other direction either, because that will basically create the same thing. Okay? Actually, we saw from the data that you cannot distinguish between a model like this, or a model like this, or a model like this, with all the different symmetries. Okay? All these models are completely equivalent from the perspective of data. The only one that is a bit different is basically this one. Okay? And this tells you that z is not the cause of x or ui. Okay? And that's the only possible wiring and direction on your graph, which will not create some correlation between x and y. Okay?
And with mutual information, because with
And-
mutual information, so when you calculate mutual information between x and z, then z and y.
Yeah, so in that case, for mutual information, this will be 0 because they are uncorrelated. And this guy will be strictly non-zero. Okay?
Yeah, so mutual information between z and y is also.
Yeah, and same for UI.